In a normal healthy state, the immune system protects an individual from environmental agents by eliminating harmful foreign substances, including pathogenic organisms, bacteria, viruses, and fungi. The main structures that participate in the immune response are the lymphatic organs, the thymus gland, the spleen, lymph nodes, and lymph nodules located in regions of frequent exposure to microorganisms or foreign materials, as in the skin, digestive system, respiratory system, and urinary bladder, forming what is called the MALT, mucosa-associated lymphoid tissue. Basically, any immune response has two phases. First, recognition of the pathogen or foreign material, and second, a reaction against it to eliminate it. The types of reaction that can be generated are often divided into two categories: innate and adaptive immune responses. Both are dependent on the activities of white blood cells or leukocytes. Innate immunity involves leukocytes that act in a non-specific manner and that respond to danger signals such as soluble mediators like interleukins or cytokines from macrophages, leukocytes, or other cells that recognize microorganisms and their products by binding to a variety of foreign molecules and then proceeding to their ingestion, kill, and degradation. Basically, it is the first line of defense while the adaptive response is initiated. The adaptive immune response depends on lymphocytes, B cells and T cells that can provide lifelong immunity. Foreign material that stimulates an adaptive immune response is called an immunogen or antigen. B cells represent 5 to 10% of the lymphocytes in the blood. They are generated in the bone marrow and are transported by the blood into the lymphatic organs with the exception of the thymus. B cells combat foreign material through the interaction of highly specific cell surface molecules called immunoglobulins, which specifically recognize foreign molecular shapes on the surface of antigens called epitopes. Once the B cell is activated through the interaction between immunoglobulins and antigens, it starts to divide and differentiate, and some develop into memory cells, which generate much faster and potent responses than when the body was firstly exposed to the antigen. Others differentiate into plasma cells, responsible to secrete large amounts of the specific antibody, which will bind to the specific epitope that stimulated their production in order to eradicate the antigen. This process is known as humoral immunity. T-cells represent 65 to 75% of the lymphocytes in the blood. They are derived from precursors from the bone marrow, which penetrate into the blood vessels and then are taken away to the thymus, where they differentiate into T-cells. That's why the name. Sooner, T-cells can proliferate and occupy other lymphatic organs. The cellular response is mediated by T-cells, and the reactions of T-cells in the adaptive immune response are generally referred to as cell-mediated immunity. Antigens are processed, carried, and presented to lymphocytes by specialized cells found in the majority of the organs, which include heterogeneous populations such as immunostimulatory dendritic cells, macrophages, and B cells. Such specialized cells are known as antigen-presenting cells. In the processing stage, proteins are partially digested into small peptides with 8 to 12 amino acids in length, which bind to an MHC molecule. T-cells cannot recognize antigens without such processing. These cells recognize epitopes derived from antigens via membrane-bound proteins called T-cell receptors, which are structurally related to immunoglobulins. However, they do not recognize and bind epitopes on antigens directly, as it happens with B cells, but instead recognize short peptide epitopes that are bound to MHC molecules on the surface of other cells. Two subpopulations of T-cells exist, the helper T-cells and the cytotoxic T-cells. Helper T-cells express a marker that is used to identify them, called CD4, often called CD4 T-cells. The marker that is used to recognize cytotoxic T-cells is called CD8 or CD8 T-cells. Helper T-cells or CD4 T-cells recognize peptide epitopes presented by MHC class II molecules, and cytotoxic or CD8 T-cells recognize peptide epitopes presented by MHC class I molecules. All cells in the body, except red blood cells, express MHC class I molecules. Therefore, any cell can present a peptide epitope in association with a MHC class I molecule for the respective CD8 T-cell to recognize. MHC class II molecules are only normally expressed on the surface of antigen-presenting cells, such as dendritic cells, macrophages, and B cells. These cells present peptide epitopes in association with MHC class II molecules for the respective CD4 T-cells to recognize. If the MHC is altered by the binding of a foreign peptide epitope, the molecular shape is altered, enabling the T-cell receptor to bind tightly, stimulating a response. When allogenic tissues or organs are transplanted, the allogenic cells activate the recipient T-cells and initiate an immune response. The MHC proteins that exist on the surface of allogenic cells are responsible for stimulating T-cells and bringing about rejection. Mature T-cells recognize altered self. An MHC molecule that differs by a few amino acids appears like altered self. The recipient CD4 T-cells are stimulated by antigen-presenting cells such as specialized immunostimulatory dendritic cells expressing MHC class II molecules, and recipient CD8 T-cells must recognize the allogenic MHC class I proteins plus receive the appropriate chemical signal in the form of a cytokine called interleukin-2, produced by an activated CD4 T-cell. The effector cells then recognize the allogenic MHC proteins and exert their effects on the cells expressing them. CD8 T-cells kill the foreign cells expressing MHC class I, and CD4 T-cells recognize allogenic APCs expressing allogenic MHC class II and produce cytokines that activate macrophages to destroy the foreign tissue by producing free oxygen, radicals, and enzymes. Finally, the grafted tissue becomes necrotic and dies.